recording in this session. All right, good morning. Welcome to the third day of our mini colloquium on out of equilibrium physics in biology. Glad to see you all here. My name is Timon Idema. I'm TU Delft in the Netherlands. This morning, as the other two mornings, we will have two sessions with each first an invited speaker and then two contributed talks. Combining of total length of talks of 1 hour and some time for discussions added to that. We had a change in the schedule from the original plan, which means that we have this morning we have Beatrice speaking. She's currently in Madrid, but actually just moved there. She just told me from the Sorbonne in Paris, and the work that she's going to present is going to be the work that she did there. And she spent quite some time in Paris. I think you were there for 6 years, you just said. So I guess the work that you're going to present is more of the last couple of years. So Beatrice, if you can share your screen.
Yes.
So the way we're going to do it is, of course, we'll have Beatrice's talk. Then we'll have about 5 minutes for discussion. If you want to ask a question, please do virtually raise your hand or send a general message in the chat, and then I will, as usual in the conference, ask people to ask their question. You can then switch on your mic and camera so that you can actually interact directly with the speaker. All right, Beatrice, the virtual floor is yours.
Hi, good morning. First of all, thank you for the invitation and for giving me the chance of presenting it longer. As I was introduced, I did this work mostly for the time I was in Paris. Now I moved to Madrid, where I'm continuing with this project. So the work I'm presenting, what we basically did was a large-scale analysis in which we saw, well, large-scale analysis of all the structures of proteins that we have currently in the protein database, in which we saw that the interactions between proteins and other partners tend to cover mostly the same amino acids that we can identify as disorder in other alternative structures. Okay, I will begin the presentation with a brief introduction about disorder in proteins. Later, I will explain the data analysis, how we combine the experimental data, and later I will move to the results. Okay, so I start with the introduction. I suppose not everybody is familiar with proteins. Proteins are linear chains of amino acids that fold more or less in an hierarchical way, first in quite recognizable structures that fold, forming well-defined structures that we don't know how to predict, but the protein does. And later these proteins can combine with other proteins to form bigger and larger complexes. That is what is known as quaternary structure. Okay, this works for most of the proteins. They fold, and once they have and it's precisely their three-dimensional structure in which they fold that determines the biological function of the protein. Okay, has been tested, validated experimentally in a very large number of experiments. I mean, nowadays we can, well, nowadays for the last 20 years, there have been measured structures for many proteins, and normally the most common experimental setup is through X-ray diffraction experiments, in which these proteins are growth in crystals that are repeated, in which the protein is repeated in different units, and using the diffraction patterns, you can just refine the data in an atomistic model so that one can extract the three-dimensional structure of the protein. As I said, there have been an exponentially growing number of structures available, and thanks and lucky for us, all them they have been stored in the protein data bank. So nowadays we have, like I saw here, I don't know, like more than almost 2,000 structures that we can try to analyze. Okay, so the problem is that as more and more structures they have been resolved, it has become clear that not all the proteins really follow this structure function protein, because either they do not get a structure, or either totally or partially. And for instance, if we talk about the X-ray crystallography experiments, there can be that there are certain parts or even the whole protein that cannot be resolved in the experiment. We know that there are some amino acids, I mean, we know the chain of the protein, the amino acid chain, but we know that certain part of the protein, we have no idea where the atoms are. These residues, these amino acids, are called missing residues. When they are missing in the experiment, you know that they are there, but you don't know where. Nowadays, we know that actually there is all the whole spectrum in between completely ordered, globular proteins, and completely disordered, and all the spectrum in between exists, in which it can be half ordered, half disordered, or small regions and larger regions that are disordered. Okay, for a long time, intrinsically disordered proteins, they were thought to be a rarity among the proteins, but nowadays it's known that they represent almost half of the eukaryotic proteins are largely disordered. They are fairly common, and also they are quite important for the research, especially because they are pathologically related with severe illnesses. I'm thinking of diabetes, Parkinson, or cancer. For instance, the 80% of the human cancer associated proteins are largely disordered. Here I saw, for instance, an example of the protein P53 that can only be resolved in a structure, can only be resolved in, for instance, this is NMR experiments. Because if you try to do it in X-ray, you can only obtain the structure of a small part of this protein. Okay, with this I pass to the definitions in the data analysis. As I said, we did a large-scale data analysis of all the crystallography structures that were available last year. From then, okay, many of these structures, they correspond to complexes. That means they have several proteins in it. We split all the protein chains. So as if we had 134,000 complexes, when we go to chains, we will have like 3 times more. And for each chain, we compute which amino acids they were part of the interface with other partners, which amino acids they were missing, they were lacking in the structure, and which amino acids they were what we call soft disorder, that I will define later. So we are going to have a chain in which, for instance, here I mark as interface in blue, or orange, it will be the soft disorder, and yellow the missing. Here I saw the binding sites. Okay, so we analyze interface that can be between proteins, protein-protein contacts, or we also consider protein contacts with DNA or RNA. Okay, so the missing residues, as I said, they appear in the experimental information. You know that, for instance, the first residue is missing in the structure, and we define it as soft disorder as the amino acids in which we know that the experiment has problem to resolve the structure. This can be measured with the Debye-Wall error factor that in biology is normally called the B factor. That tells us how well you're determining the structure of the position of a given atom. Okay, having had B factor can tell us that this region is highly flexible. For instance, it fluctuates in time, but it can also mean that the atomistic refinement model in the X-ray diffraction experiment is failing. This could mean that, for instance, the material is amorphous, it's rigid, but it's not reproducible in the crystal, like if it were a glass. So with high B factor, we are gonna identify these both situations that we cannot distinguish, but it can be either flexible or amorphous. Okay, experiments, they are done in different, the available experiments, they are done with different resolutions. So we need to be able to normalize the results so that we can we can mix data from different experiments. And what we did is to select as soft disorder the residues that would have a B factor that would be on the Q of the large B factor. That means that we selected all those that they have B factor that are higher than the mean plus the one standard deviation. Okay, this threshold, it's not very important. We have studied also the thresholds, and if you're interested, I can explain more, but it's not very critical for the analysis. Okay, with the choice, we have like, we are selecting like 16% of the whole chain as disorder, and we will analyze it. I will explain it later. Okay, so once we have all these 3 possible flags for the amino acids, it can be interface missing of soft disorder, we are going to cross all this data, obtain it with alternative crystals in which a protein is present. Okay, as I said, we have different protein complexes in which we can have a protein interacting with different partners, or it can be this, we call it bound, or it can be the protein all alone that will be unbound. Okay, to build these clusters, we need to, what we did was to try to find in a recursive way all the chains that contained all the structures that contain in a protein with a particular amino acid chains up to a sequence identity of 90%, and we also selected chains that are more or less of the same length. In total, we have like 47,000 clusters that we will analyze it. We will combine it to obtain the statistical analysis. Okay, so we have a cluster. Here I saw an example in which it has 45 entries from the PDB, and it contains different chains of different complexes. We need to give a name to its cluster, so we selected automatically the name of one of the structures, the one that had better quality, either better resolution or other criteria. And okay, once we have the cluster, we are going to align all the sequences to the representative cluster. In general, they have the same sequence, but it could have some amino acids that may be longer or maybe shorter or some gaps. But what we do is in the same sequence, we tag for each of the structures in the cluster. We will tag where it was interface, where it was disordered or it was missing. And later we compute the union of the information of all the clusters, so that we can see which amino acids they were interfaced at certain moment in the PDB, and all the amino acids that were disordered at certain moment in the PDB. Okay, for instance, I saw here an example of a cluster. As I said, they had 45 structures, but okay, we identified 12 different possible interfaces. And we can also identify the different disorder regions here. Normally we do it in the sequence. Here I just plot it in the protein structure. And the nice thing is that once we take the union of all the experimental information, we see that the disorder regions, which are the orange, they normally involve the same amino acids that are involved in the interface, in the union of all the interaction interfaces with other partners. Well, here I also saw an unbound. That means that there is no interacting partner. This is not very important, but I will use it later to distinguish it from the bound, the situations in which the protein is interacting with partners. Okay, so I pass to the results. I start with comparing the typical size of the disorder regions and the interface region. Here I saw the relative size in the sequence in blue we have the interfaces, in orange we have the disordered regions as function of the number of different interfaces that we observe in the cluster. And we saw that both kind of measures, they follow very similar statistics. Also, we can compare the number of connected clusters that we observe in the structure, and we see that they behave more or less in the same way. As the number of interfaces grows, we start to observe different connected regions that grows with the number of interfaces, and at a certain moment, all the different regions start to percolate, so we just observe a single region. So we lost the signal, but we lost it in the same way for the interfaces and the disordered regions. And also coming back to the other figure, one nice feature that we observe is that we can see that the relative size grows essentially with the logarithms, linearly with the logarithm of the number of interfaces, with suggests that the interface, the different interaction interfaces are gonna organize in an hierarchical way. Just to explain what I mean, we expect to have a bound structure, later have simple interfaces of the protein with one partner or two partners in different regions, and later we expect to find other complexes in which the same simple complex appear interacting with an additional partner or even with two additional partners. That's what we expect from this logarithm is a growth of the size of the regions with the number of interfaces, because if it were not the case, we would expect that it grows linearly with the number of structures. Okay, we can try to explore this in our clusters, and actually that's what we see. Here I saw an example that we did with the protein p53 that I showed in the introduction. For instance, here we have the unbound structure. We can find structures with this protein interacting with DNA. Here I saw the interface with DNA in green. Later we can find other structures in which we have the same interaction with the DNA, but also an additional interaction with a protein, and so on. The same complex here, but interacting with an additional protein down there. Okay, we observe the same behavior for the disordered regions, and I will try to argue later that actually the sub-disorder is also related to this hierarchical growth of the complexity of the protein interaction network. Okay, so as I said, the disordered regions, once we average it over all the clusters that we have, over all our 47,000 clusters, we observe that both kind of regions, the interface and the disorder, they behave in a very similar way. The direct question is if they actually spot the same regions. Here I saw several examples with different number of interfaces in the cluster, and well, visually they look very much alike, but we have like so many possible clusters. So if we want to do a statistical analysis, we need to compute. For instance, we did it with the median of all the clusters. Okay, so the idea is that we are going to compare where we find, to see if where we find a disorder region, if we find also an interface region. And for instance, we can compute this positive predictive value, which is the probability that if I am orange, I am also blue. And what we see is, okay, we have the different symbols here correspond to the different structure that I saw here. In orange, we have the median of all the values that we obtained in the cluster. And in black, we have the expectation for a random location of this disorder amino acid. Okay, what we see is that being disordered, it's at certain step predictive of being also interface. They are correlated, but still, I mean, we get an improvement over random, but not an incredible improvement. Okay, this we know that disorder tends to be interface, but we can go further and to see they are really involving the same amino acids. For this, we can use the rock curves, and to show here on the vertical axis, we saw the true positive rate versus the false positive rate. Okay, if there is no connection between them, we would expect that the probability of being a false positive or the probability of being a true positive, it will be the same, so we will have the random situation. Okay, what we see is that here in this picture, I analyze all the clusters by selecting them in the basis of the number of interfaces that I detect in the cluster. And when we just have one interface in the cluster, we see that the connection between the disorder regions and the interface region is essentially worse than random. Okay, the better the predictor is, the larger is the area below this curve. If the connection is perfect, we should expect a line, an horizontal line here. If it's very bad, we would expect like random, or if it's worse than random, we will go below. Okay, so for one interface, as I say, we are essentially random or even worse. But as we start to select only the clusters that have certain number of interfaces, not that many. Here I think it was the 8 interfaces. We start to see that the connection between them, it starts to be quite much better than random. Still, this is not a good predictor yet, unless we have too many interfaces. So we see that there is a correlation. So we see that they tend to be located at the same, more or less, amino acids. But as I said, we don't get a perfect connection between both. So I just want to stress that in any case, even if there were a perfect connection between the being disorder and being interface, we couldn't see it because the protein data bank is very, very incomplete. We don't know all the interactions of a given protein. We just have a subset. So if there were a connection, we couldn't see it. We expect to have a lot more false positive than what it will be in the real nature. So the question is how we can test that this is the case, that we have information that is missing. One situation that we can clearly approach here is the unbound forms. We can select clusters in which we do have the protein alone. We have a structure in which the protein doesn't interact with other partners, and separated with the clusters in which this situation is not there. What we see is that when we analyze clusters that have at least one unbound form, this was the figure I was showing before, we see that already from one interface, we start to get some predictive power of the disorder parts, which was not the case when we were obtaining it all. And in general, we can see that we get much better performance if the unbound structures were present. So this is telling us that if we know the disorder regions in the unbound, we managed to cover better the union of all the interfaces that if we didn't know. This suggests us that because in general one doesn't see that in a given structure the interface is disordered and interface at the same time, because normally the interface is quite rigid. So this is telling us or suggesting us that that the disordered regions could spot the interfaces at the next step of the assembly. Okay, you might not understand me at this point, but you will see later in some examples. Okay, here I saw this hierarchical growth. So the idea is to try to check if this behavior of the disorder, if disorder could predict us which will be the position of the new interfaces in the hierarchical graph. Okay, so this we can check it in particular examples. I saw, I think I go through the branch I was showing before. Okay, when we get the unbound structure, we can check where the disordered regions are, and we can see different spots that actually correspond to a not very precise, but quite good spot, quite good way, the location of the interfaces that we observe when this protein is just interacting with some additional partner. Okay, later in this structure, we can check where the disordered regions are. In this case, not that clear, but okay, we observe a certain spot in which we find another interacting partner. But what is more interesting is that, for instance, when we take this structure, if we analyze where the disordered regions are, they completely change from the location where they were up to now. And in this case, we start to observe it below. And in fact, we observe other interfaces in higher order interaction networks of the protein at the same region. So this suggests us that the protein disorder is controlling the way the protein is assembled in complexes. Okay, here I saw another example of the same protein. For instance, here after binding with a protein on the top, we start to observe a region that was not before disorder, in which we also find other interfaces in which the same complex is repeated. Okay, here we will have this, this, and this, and we have additional part. Okay, so this suggests us the idea that disorder could be controlling the protein assembly process. And here we analyze different complexes in which we know which is the assembly path in nature. There are not many cases in which we have all the structure resolved in between. Here I saw one in which we see different disorder parts. Is the same protein that combines with the same protein, forming an interface here. Later we have two of them assembling here. And once we have the four protein complex, we start to observe other regions which are disordered, in which we also observe the new interface. With this was one case, and another case we have a dimer in which we can observe really well determined when we have the dimer alone, two well-defined regions that actually are quite the same region at which we find interfaces once this dimer is interacting with another dimer of the same kind, forming a D2 structure. Okay, so this suggests that actually disorder might be telling us where the next partner will be coupled. And okay, these are preliminary results, but now we have been trying to analyze this in a statistical way. Okay, we have all these clusters. We can build hierarchical graphs of interfaces, as I saw before, and later we can average over of the clusters and to see if the disorder at a given node can be predictive of the interfaces as the next node in the graph. And as I saw here, not very clear, but what we see is that actually the predictive power works better than when we zoom up of the structure. Okay, we get a clear signal that it even gets better when we get the larger is the complex, the more direct is the correlation between the position of the nest structure. And with this, I finish. Okay, we analyzed all the crystallographic structure, and we saw that the disorder regions tend to cover the same amino acids that the interfaces regions, which are telling us that disorder seems to play a role in determining how the proteins are going to be assembled. In the way that it's quite intuitive that this soft disorder might be highlighting the regions that are a bit soft, so that the interface can be located easily, which is a bit different picture than the traditional lock and key scenario in which you have two perfect proteins with the correct structure to match themselves. Here what we are saying is that interfaces are going to appear in regions that are quite soft. And just I finished in that, okay, I recently, as I said, I moved to Madrid and I have funding for a postdoc of 3 years. I'm looking for candidates to try to apply statistical physics tools to try to understand better protein disorders. So if someone is interested, please contact me. Okay, so thank you very much.
All right, on that appealing note, thank you for your talk. As I said, if you have, and thank you for staying on time, actually well within time, so we have time for multiple questions. If you have a question, please virtually raise your hand or send a chat message. So yeah, so there we go, Juan.
Nice talk. I was wondering if somehow you can predict how this, because if I understood correctly, the clustering of two proteins triggers the release of another subregion in another protein. So can you correlate this with how the stresses are propagated through the protein? So I mean, if I know that these two proteins are connecting in this region, can I predict which region is going to get softer?
I have no idea at this point, but I mean, it has to be. I mean, it has to be this way with it propagates because it's changing the structure somewhere distant. Yeah, I mean, I don't know. I mean, there are quite a lot of people working on
I mean,
changing the proteins here and how it changes the structure there. But I don't at this moment. I just analyze the experimental structures. I don't know.
Yeah, but now you can put information about the constant spring of alpha helix and beta sheet and see if there is a path easier or using your data. So it's just feeding another set, right?
Yes. I mean, now that we have quite a robust database, there are quite a lot of studies that can be done. Because, yeah, sure.
Thank you.
All right, thank you. So there's two questions in the chat. One is very easy. If you can go back to the last slide so that people can copy the reference to your work,
okay.
Which is now down here, the bioarchive. And the other one is from Oscar Morera. Oscar, would you like to ask it in person? It's more interactive.
Hello?
Hi.
Okay, hi. Could I ask you what the percentage of disordered proteins are there in an healthy person?
What? Sorry?
How many disordered proteins can be found in a healthy person?
In a healthy person?
Yes.
Well, I mean, it's not that the protein disorder means that you are not healthy. I mean, as I said, statistically, more or less, we have in the eukaryotic kingdom in general, it has around 40 or 50% of it. I mean, our proteins, a lot of them, they are disordered. It doesn't mean, I mean, and there are different theories of why illnesses can be related to disorder proteins, but the problem is that in general, disorder proteins, the more disorder is a protein, the more partners it has. So the more functions it's going to have. So if there is something malfunctioning, it's going to trigger a lot more possible problems than if the protein is ordered that tend to have less functions. But no, I don't know how to answer the question.
Okay, thank you.
All right, are there other questions?
I can ask another one. Is this interaction reversible in the sense that if two proteins aggregate on one side, triggers the softness of another, but if I remove this one, this attach too? Or
I didn't try to look at the cycles in this way, but actually we see more or less this, because a lot of time, I mean, these graphs are not tree-like. I mean, we can have a structure that can be arrived from different, for instance, here, we can arrive in our construction to this structure either coming from here or coming from here. And we do not observe in general that the disordered parts are the same in both of them. I mean, it seems really, once you pick, you frozen some part of the protein, you're triggering another part. I would say that it is, but I mean,
okay.
I mean, with the examples I saw, it seems to be, but I cannot say that it's general.
Okay, thank you.
So I will use the chair's prerogative to ask my own question. So you were showing some examples of dimerization of proteins.
Of what? Sorry, examples of?
Of dimerization, where two proteins, two identical proteins coming together to make a protein dimer. So I was wondering, so because of you're trying to correlate the disordered regions to interfaces, does this affect the specificity of these connections? Do you expect that if two things really need to dimerize to function, you get less disorder or more disorder, or is this not something that would have an influence?
I mean, one of the things that we want to see is something that disorder can specify which partner you can have this. I don't know if this is the case. We will study it. But I think that this has to, I mean, that actually it specifies at least the region, because sometimes we see that there are different kind of, I mean, we have many examples in which the disorder spot the region in which we can find interfaces, but not always is the same partner that is there.
All right.
But yes, about the specificity, it's something, I mean, we were trying, for instance, to see if we could find some kind of different behavior if the partner was DNA or if it was protein. It's a specificity, not very specific, but at least more, and we were not able. This, no.
Okay, there's also no distinction even.
They might be, but I don't, up to now, I didn't manage a way to find it without introducing other bioinformatic predictors. I mean.
All right, then if people have more questions or want to know more, they can look up your, we can contact you for a job or for more information. Thank you very much for your talk. I would like to ask you to stop sharing your screen so that our next speaker, who has already switched on his camera, good morning, Rodrigo.
Good morning.
Can start. Yes, you should have been made co-host so that you can share your screen. Yeah.
Let me see. So, can you see my screen?
Yes. So our second speaker is Rodrigo. I hope I will pronounce this not too awfully. Coelho.
Yes, it is
correct. From the University in Lisbon. Talking to us about also interfaces, but now not in proteins, but in active liquid crystals. And you have 15 minutes for your talk. I will give you a private message when you have about 2 or 3 minutes left.
Thank you, and good morning, everyone. And I'd like to thank the organizers for the opportunity to present our work. So in this presentation, I'm going to show our numerical investigation on the propagation of interfaces in active liquid crystals. Well, active liquid crystals are mostly living systems, which are elongated and densely packed, and are able to convert the energy from their surroundings into directed motion. You can see examples in these figures. For instance, in this video, we see components of the cells. They are the microtubule kinesin, and they are in interface between oil and water. So because they are active and elongated, they form this chaotic flow that resembles fluid turbulence. Other examples are colonies of bacteria, which are elongated, and even macroscopic systems as shoes of fish. These name active liquid crystals come from the passive ones, which are widely used in display technologies. You are probably watching this through a liquid crystal display, and they are formed by elongated molecules that have a dual behavior between liquid and solids. So they can flow like a liquid, but they are still ordered in a crystal-like way. Their particles are aligned for a certain position. In the ordered phase, when the particles are pointing for a preferred direction, we say that they are in the nematic phase. And when they are pointing to random direction, say that they are in the isotropic phase. In our system, you have these two phases coexisting with an interface between them. And the model you use to simulate these active liquid crystals is based on two main equations. One gives the time evolution of the liquid crystal itself, the particles, of the alignment of these particles, and it's called the Residues equation, and it depends on the minimization of the Landau-de Gennes free energy. This free energy have two terms, one bulk term that gives the phase transition between nematic and isotropic. In our case, these two phases will be in the coexistence region for certain parameters, and an elastic term that penalizes distortions. We note that this Residues equation depends on the fluid velocity. So the evolution of this fluid velocity is given by the Navier-Stokes equation. If compared to the Navier-Stokes equation for normal isotropic fluid, there are three new terms. One is the stress coming from the particles of the liquid crystal. It depends on this order parameter K alpha beta. It's the same using for passive liquid crystals, for simulations of passive liquid crystals. And there are two other terms. One is the friction with the substrate, because in our case, we want to simulate bacteria which are on a substrate and subject to friction. And the active stress that's proportional to this parameter zeta, this gives the strength of the activity. In our case, this parameter will be always positive, meaning that our system is extensile, but it's also possible to have negative values for contractile systems. Our motivation for this particular work was an experimental paper from 2018. Whenever I talk about the experiment, I'm referring to this paper, where they have studied swarming bacteria of a specific type of bacteria, Serratia marcensis. And these bacteria are slightly elongated, and it's known that when they are in the swarming mode, which is a collective mode of propagation for bacteria, they are more elongated. They become more elongated and more flagellated. So their liquid crystal properties are enhanced in this swarming mode. These bacteria can be killed using ultraviolet light. So what the authors did in this experiment was shed light in a certain region. For instance, you have here the velocity field, the propagating bacteria. They shed light for a circular region approximately, and the bacteria in this region die. And they analyze the propagation of this active-passive interface. So with time, these died bacteria are being diluted in the active domain, and in the end you have only active bacteria. The died bacteria are diluted in this region. Here you see the same experiment for a flat propagating interface. You see that the died bacteria resembles a solid which is being eroded by the living bacteria. You know? And here you see the results from our model for active liquid crystals. We try to reproduce the same conditions as in the experiment. So we start with the chaotic flow, and then for a certain time we set a region of circular region of isotropic liquid crystal. Then this circular region reduces, and in the end you have only nematic in this domain. we see in the velocity field that this region of isotropic fluid has zero velocity. So we also have an active passive interface. And here are results for a flat propagating interface as in the experiment. So we studied these two shapes for an interface because they were the shapes studied in the experiment as well. And to be sure that we are in the same flow regime as in the experiment, we reproduce the statistical quantities of this system. They have measured the velocity distribution, which follows a Maxwell-Boltzmann distribution. They have measured the spatial and time correlation functions. So the spatial correlation has this minimum, and then the correlation becomes zero for a certain region. It gives the length of the average length of the vortices, for instance. And they have also measured the energy spectrum for the system. This is more interesting for us because we can compare with these two power laws. These two power laws were also found for other living systems as a colony of Bacillus subtilis. So, and there is some discussion in the literature if these power laws are universal or if they should depend on the activity. But we want to compare with these quantities, and we have measured these quantities for different activities. In the experiment, I have used only one for some restrictions, and for instance, in the spatial correlation function, we have different curves, but they all collapse in a single curve when we divide the distance by the length scale of the activity, which is given here. So it means that this is the relevant length scale, the dominant length scale for the system. Also, the time correlation function collapse when we divide by the length scale of corresponding to the activity. And the energy spectrum follows the expected power loss. So in the increasing range, you have a power law 530, and the decreasing range, it decays with minus 830. At least it's not too far from what was found in the experiment. Due to the fluctuations, it's difficult to affirm that they are strictly these two power laws. So having characterized the system, we can, using the model to make predictions about the swarming bacteria or the propagation of interface in this system. For instance, we have measured the closing time for a circular interface as a function of the activity, and we found that it decays quadratically with the activity. So with this observation could be used to develop protocols to measure the activity in living systems, if it's verified in swarming bacteria. Have you also studied the flat propagation of a flat interface? We measure the velocity of this, the average velocity of this interface, which is constant in time for different activities. Also, the interface width of this interface is constant in time, approximately constant as occurring in the experiment, and we can detect the exact position for each position x by using the order parameter. When it is between the isotropic and the kinematic, we say that the interface is in that position. And we observe that there are many oscillations also in the experiment and in the simulation. So we can take a Fourier transform from this interface position and count the oscillation modes. This is the destructive factor for this interface. In the experiment, they have compared to the equilibrium value, which follows a -2 power law. In our simulations, we have measured as well, and compared to this -2 power law, it follows approximately this. But what our simulation suggests is that it should depend on the activity. It changes slightly with this. Another prediction from our simulations is that we can stop a propagating interface with a gradient of friction. So if you have a lower friction on the bottom and higher friction on the top, and we start with a pneumatic liquid crystal, active liquid crystal, the pneumatic will form clusters in the region with higher friction, which shrinks, and in the end you have only isotropic in this region. So the interface stops for a certain position, and it still fluctuates, but the average position is constant in time. And this can be used in applications or in experiments where you want to isolate the swarming bacteria, and experimentally we can control the friction by means of the concentration of agar of the substrate. And the position where the interface stops depends on the activity. So for higher activity, it stops in a different position, and so this position gives the threshold of friction for which the interface can still propagate. So if you have a friction slightly higher than this threshold in the whole system, and you start with a pneumatic domain, it will form clusters, and in the end, we have only isotropic, so we don't have a propagating interface. And if you start with a slightly lower friction, we still have a propagating interface. So we measure this threshold of friction for each. We still have an interface as a function of the activity. So to conclude, I have presented our numerical investigation on the propagation of pneumatic isotropic interfaces in active electric crystals, which are on a substrate. And we found that this model reproduces many statistical quantities of the swarming bacteria, so we can use it to understand this system using simulations. We have found, for instance, that we can use the closing time of a circular interface to estimate or to measure the activity in living systems, and also how to stop a propagating interface with a gradient of friction. So you can find more details in our publication, and thank you all for your attention. I'm open for questions.
Thank you very much for your talk. A lot of different topics in the freedom. Are there questions from the audience? There's not one yet. I would like to ask a silly question, because I was looking at your movies and trying to make sense of what I was saying. So you have a blue phase, which is the isotropic phase, and a red one, which was had some value, some finite value of your order parameter, and then you have little fluctuations in there. Are just those just the director field, or what are we seeing?
Good question.
What is actually plotted here?
Yeah. So indeed, we have the pneumatic phase in this region, in the tropic, in the blue region. These fluctuations are the defects. So we don't see here the directors, but they form positive and negative directors. So some of them have a shape of a comet, and others have a shape of a star. So we see that we have -1/2 and +1/2 defects, and the positive defects they propagate in the system. So that's why we see these fluctuations moving. Good question.
All right, thank you. Are there other questions? So I actually had another question. Oh, there is one from Juan also. Juan, please go ahead.
Oh, really nice results. I'm just kind of curious about, because for an active system, one would expect higher fluctuations in the interface, right? Are rather small in the experiments where you have this one. Are not that big, those fluctuations, right?
Yes, these fluctuations are they are related to the size of the vortices in this region. And these vortices they are controlled by the length scale of the activity, as I mentioned there.
Yes,
that expression, as well as the length scale for the friction. I don't show here, but if you change these two quantities, the friction, the activity, the vortices, the size of the vortices change, and also change the size of this fluctuation, the amplitude of the fluctuations.
And another question: do you have any physical insight about those power laws that you see? Why those power laws?
No, it's too. There's still debate in the literature if these power laws are universal, but you have in this region of length scales for smaller wave numbers, an injection region where the liquid crystal injects ends on the system. Then here is the inertial range where we start the energy starts to decrease. So for larger wave numbers, the system starts to dissipate. So in the small scales, the small length scales, the energy, the friction dissipates, and the friction and the viscosity dissipates the energy in the system. So yeah, there is the injection range and the region where the dissipation region. Yeah.
Yeah, that's what. Okay, now I can see. I can make the image in my head. So thank you. Nice work.
You're welcome. Thank you.
Thank you. I see we have another question from Raul Marate, our next speaker.
Yeah. Hi. So just wondering whether you see more than one defects, and whether two defects at two places come close and coalesce, or a big defect separates into two or several. Is it? Do you see this kind of behavior?
Yes. Yes. Initially, we start with random directions, and with time, many defects are formed. And indeed, we have hundreds in the system, which sometimes they merge. Yes. So we have destruction and creation of defects, but the average number of defects, which was shown in previous works from Julie Yeomans, it's approximately constant in the system.
Okay.
Yeah,
thanks.
All right. Thank you very much. Very nice work indeed. Congratulations. I would like to ask you to stop sharing your screen so that we can move to the last talk before the break, which is from Raul, who very courageously asked the question in the previous talk. So you can get this back later. If you can put up your talk. So you're from the Indian Institute of Technology, right?
Yes, that's right.
So you're actually, it's late afternoon for you?
Yes, it's 2 o'clock.
Oh, it's not that bad. So, and you're going to be talking about twitching bacteria, which I guess are somewhat related to the previous talk, but also somewhat different, in ratchet geometries. So please enlighten us about that.
Okay. Thank you very much for giving me this opportunity to present my recent work. So this work has been done with my PhD student, Konak Bisht, and he has already defended his thesis and looking for postdoctoral positions. So the plan of the talk is something like this, that I will talk about the twitching motility, what I mean by twitching motility. I will show you briefly some earlier results. And I will then put this bacteria into corrugated or ratchet geometries and try to see if we get a directed motion. I will discuss the results and then I will conclude. Okay, so twitching motility is one of the several forms of motility many bacteria show. And this is usually seen in human pathogens, for example, like Neisseria gonorrhoeae. And this is especially related to the bacteria that tend to move on 2D surfaces. Okay, for example, human skin or internal organs, etc. So this is a very special kind of motion in which a bacterial cell body produces what are a polymeric structure. You can see this in a figure. The blue blob, the yellow blob at the center of the figure is the bacterial body. And
I'm going to interrupt you. What I'm seeing is still your opening slide, and I think this is probably true for
me. Oh,
really? And I think you have something else on your screen. So maybe you have shared the wrong screen. Sorry for interrupting.
Okay, sorry. So I will stop sharing and I will again share.
Yeah, please do.
Sorry for that.
That's okay. That's why I'm here. Yes, now I see a yellow blob that you were talking about.
Okay. So there is a yellow blob at the center of the figure, and that's the bacterial body. And this yellow streaks which are coming out of the bacterial body are these polymeric structures. These are called as pili, and a singular is pilus. So these polymer structures come out of the bacterial body, adhere to the surface, and then they start retracting so that the bacterial body basically moves forward. So if you think of this as there are several pili which are involved in this kind of a motion, and you can think of this motion as a stochastic two-dimensional tug of war. And this is how the bacteria moves on a surface. Okay. So it is interesting to know that a single pilus, which is this polymer, if it is attached and tries to pull this bacteria, it produces the forces of the order of 100 piconewtons. And this is the strongest motor that you have in biological systems. The experiments are performed to see if the bacteria shows any persistent motion. Persistent, what I mean by persistent is the directional motion. And it's not just random motion that this bacteria perform. So if you look at a wild type bacteria, then the average persistent time is of the order of few seconds. And the experimentalists can mutate these bacteria to produce more or less number of pili. And if the number of pili are more, that means it's getting pulled in all possible directions with more number of pili, then this persistent time or directionality of the bacteria increases. It also knows if you pull, for example, this pilus which is attached to the surface, then only a force of 10 piconewton is required to detach it from the surface. And if you compare the forces generated by a pilus to pull a bacterial body is 100 piconewtons, and it's one order small to detach it from the surface, the pilus from the surface. And these are the basic conditions required for a two-dimensional tug of war. So here I show a cartoon of what happens. So in figure A, what you see is a blue circle, which is the bacterial cell body, and the lines which are coming out of the cell body are these pilus. I assume that these pilus are straight lines, and they are of various length, and they are attached to the surfaces. Only attached pilus pili take part in the motion. And as you can see that there are 4 pili in the third quadrant of this figure, and hence there will be more force this bacteria will experience, and it will basically the move in this, let's say, a 7 o'clock direction. However, this number of pilus which are present at a given time instant is a stochastic process. So it may happen that a new pilus may be created. The existing pilus can detach from the surface and so on. As soon as these processes happen, the bacterial force balance that is pulling the bacteria gets changed, and the bacteria can actually change the direction depending on where it experiences the maximum force. And that's why I call this motion a two-dimensional tug of war. This kind of motility is also known as twitching motility, as I said earlier. Figure B shows a process in which the directional change of this bacteria as it is moving may happen, and the details of the model, etc., can be found on the in the reference which is below. So there are several important parameters that we require to model this. One is the pilus creation rate. So we know that in a wild type bacteria, on an average, there are these 7 pili which exist. This is again average. So in a given instant, there can be less or more pili than this. Then we should know what is the pilus detachment rate as a function of the force it is being pulled with. Then we should also know pilus retraction and elongation as it extends or elongates or retracts as a function of the force it is being pulled, as well as the average velocity.
Oh, you have
several
lost
patches.
your sound.
Oh,
you're back. Thank you.
Okay.
So last thing I heard was you were talking about the average velocity as so the penultimate bullet point. So you're at the average step length.
Now, am I audible?
Yes, you are.
Yeah. Okay. So in figure C, if you see, there is a bacterial trajectory which I have shown, and there are several regions where you can see that the bacteria actually goes in a directional fashion, and some regions which are very squeezed, there it tries to do a kind of a random walk. So if I take such trajectories and try to coarse grain it, then what I will see is a bacteria which chooses a random direction in 0 to 2 pi uniformly and takes an exponential step. So now what I have are two models, which is the one is the detailed twitching motility model, which I have represented in this figure. And the next, a coarse grain model, where it's like a run and tumble, where the bacteria follows an exponential step length distribution, which is observed experimentally. And each step starts with a randomly chosen direction uniformly between 0 to 2 pi. And a bacteria takes a step length from an exponential distribution and moves there with a constant velocity 1.5 micrometer per second, which is also known experimentally. So now we try to compare these two models, the detailed tug of war model and a coarse grain run and tumble motion, and we try to see what happens in case of its motion qualitatively as well as quantitatively. Okay. So now we consider these two models, a detailed tug of war and a run and tumble motion, and we try to put this twitching bacteria in a corrugated channel. So this I have shown in the figure which is on the top. So this is a corrugated or a ratchet channel which is asymmetric, and the boundary function is given by a function of sinusoidal harmonics, which is on the right hand side. So you will see that this channel is asymmetric, and there is a preferred direction that this bacteria shows. So this spatial asymmetry gives rise to rectification of this twitching bacteria. Note that unlike other bacteria, for example, E. coli, this Neisseria gonorrhoeae are not chemotactic, so they cannot detect a chemical gradient. But it is seen experimentally that their motion is definitely affected by the surface topography. As you know, Neisseria gonorrhoeae is a deadly pathogen, and it tries to live on human skins and internal organs. And of course, its motion is expected to be affected by on which tissue it's living. So there are several studies which take care or which study this kind of ratchet systems, and these are known as active ratchets. Unlike Brownian ratchets, where these ratchet potentials are switched on and off, and thus the system is driven into the non-equilibrium, and you see a directed motion. There is no switching on and off or flashing of this ratchet potential. The activity of the bacteria itself drives the system to non-equilibrium, and asymmetry plays a role in directing its motion. So the activity, what I mean by activity is bacteria or Janus particles, they use some kind of energy to propel themselves. Bacteria use ATP, and Janus particles probably some light source which is being operated from outside, some chemical energy. And because of this activity, it is essentially a non-equilibrium system. To check if this switching bacteria actually rectifies in this asymmetric motion, what we look at is calculate average displacement, and which is given in this, which is x minus the average initial condition. And then we actually see that there is a rectification in the motion in both models. So here I have, for comparison, put two parameters. One is RCR, is the pilot's creation rate, and LP is the average step length, and they are basically related to each other. The average creation rate will tell me the average number of pili which are present in a tug of war model, and LP will tell me the average step length, how far it goes when it takes a step length on an average. And as t tends to very large, you see that delta x is actually not 0 as would be seen in a normal Brownian motion, but it has a directed motion. So the next, what we do is we also look at mean square displacement and the square displacement. And what you see is that for long time, the motion is ballistic, and then it goes to a diffusive motion. Same thing happens for a coarse grain model. One thing is to note that as said earlier, that for a wild type bacteria, the persistent time or the ballistic motion times are of the order of few seconds, 1 or 2 seconds. But in case of ratchet geometries, when I put this bacteria in ratchet geometries, they are of the order of 10 seconds or so. So the persistent of this bacteria is highly enhanced when one looks at asymmetric corrugated channels. So next, we also plot the average scaled spatial probability density of finding the particle in a given channel. And one can see that most of the time bacteria tends to remain at the boundary, and the motion or the directed motion is actually a boundary driven phenomena. So the details of this model and simulations can be found out in the Physical Review e-paper, which is just published in 2020, and the reference is at the bottom. Okay. Now what we do is we try to see what happens to the particle current along x direction. So Jx is the average velocity divided by the time in the long time limit. And this negative sign is just to make sure that everything is correct, because we have chosen the asymmetry of the channel to be in the negative x direction. So this minus sign is just there because of without loss of generality. And what you see as you keep on increasing the creation rate or the step length of this bacteria in both these models, the current actually increases and then it saturates. The saturation is expected because once the particle starts taking jumps which are much larger than this ratchet cell size, it will get stuck in the given cell, and it will be very difficult for it to move from cell to cell. Thus, it gets saturated. We also see that how current behaves as a function of the ratchet parameters or the channel parameters, namely the minimum width and the maximum width in this asymmetry. And what you see that you can always choose a parameter where the current can be optimized, as can be seen from figure A. As Wmax increases, the current actually increases, reaches a maximum, and then it starts decreasing. because as W max increases, W mean also changes, and it becomes almost comparable in size. So it's as good as a straight channel. And similar things are also seen in a run and tumble motion, as well as in the detailed model. So the take-home message from this figure is that the current can be optimized by adjusting the ratchet parameters. Okay, so I will stop here. I will conclude with this. The rectification of twitching bacteria is studied in a corrugated channel using a tug of war model, which we established some years ago, and a run and tumble kind of a generic model, which is used for several active particles. Unlike our earlier studies where hydrodynamic interactions are very important for a directed motion, here the interactions are only steric because the particle is, or the bacteria is moving on a surface. Asymmetry of the channel, of course, plays very important role in rectification, and channel parameters can be chosen to optimize this motion. Qualitative features in detailed tug of war model and coarse grain model remain the same, but quantitatively they differ in terms of persistent time, etc. So with this, I would like to thank you, and I'm happy to answer your questions. Thank you very much.
Thank you very much. Thanks for being flexible, and technology was say preventing you from speaking. We have a question already here from Miguel Garcia. Please go ahead.
Yeah, hello. Thank you very much.
Hello.
So I have a, maybe it is a naive question. Like, can you go to the beginning when you explained like the forces
Okay.
that
Sure.
I think it was like 100 piconewtons of pulling and kind of 10 piconewtons to detach it.
Right.
It may be
Is
very
this,
naive, but
okay.
is it because they have different directions? Like, why doesn't it get, when it is pulling like 100 piconewtons, if it is only 10 piconewtons to detach?
No.
How does it
So
work?
no. So what is happening is you have a cell body, and a pilus is coming out of it, right? And it gets attached to the surface, okay? And there is a motor which is involved. So you can see in this figure that there is a bacterial cell body, and there is a motor which tries to elongate this this pilus. It gets attached, and there is another motor which comes in the body, and it starts retracting. So as it starts retracting, the length of the pilus is getting smaller and smaller, and the cell body is going forward, right?
Yes.
But now it may happen that this a pilus in this direction gets pulled by several pili in the other direction, right? So rather, now the force what is being pulled, the pilus in the other direction is being pulled at the place where it is attached.
Yes.
Right. So when it gets pulled, the pilus gets detached from the surface and not from the cell body. It remains as it is, but now it's hanging.
Yeah.
So this is how I'm comparing these two forces. So with a very small force, you can detach it from the surface because of the other pilus. And if I look at the force with which it pulls the bacterial body, is of the order of 100 piconewtons.
Yeah, I mean, I understood that you are looking at the balance between pilus that are in two directions.
Yeah.
But it may be that I'm missing something. I just don't understand if it only takes 10 piconewtons to detach.
No, okay, so there are two things. What is the maximum force a pilus can produce? It's of the order of 100 piconewtons. And so as a function of force, if I look at binding times, the amount of time it requires for a pilus to remain attached as a function of force. Okay, so for 10 piconewtons, it's already something like 10 to the power minus 2 seconds or so. For smaller forces, it will remain attached to the surface more.
Okay, so they are not all the time pulling at the maximum force. Okay, thank you very much. Okay, now I understand. Thank you.
All right, thank you. I see we have another question from Juan, but also we are running at the edge of our time. Fortunately, these two things are not mutually exclusive because the next thing on the schedule is a break.
Okay.
So before I ask Juan to ask his question, I would like to first thank the speakers from this morning's session, or for the first part of this morning's session. That's our three speakers. Announce that we have a break now, and we will continue with the next three talks at 11:10, concluding with a talk with someone comes from, say, also from far away, but then the other direction. So they are getting up very early for us. So it's nice to have people from all over the world in our session. So thank you very much for your talk and for your presence. We have a 20-minute break, and Juan, I would very much like to allow you to ask your question now. Everyone else can go get coffee.
Juan, you can go now.
Okay, so we are going to resume this session. And same as before, you can please write in the chat that you have a question, and I will indicate you when you can open your mic to ask yourself the question. And so let's continue with our next speaker, that is Jeonju Baek from Seoul National University. And he's going to talk about generic symmetry breaking motility of an object in an active fluid. So the floor is yours, Jeonju.
Thank you very much. So hello, I'm Jeonju Baek from Seoul National University in South Korea. Today I'm going to give a first I would like to thank the organizers for giving me this wonderful opportunity to talk in this conference. And this talk is going to be about my recent work, which is still ongoing with my students Ki-won Kim and Yoon-sik Choi. So I must first warn you that this is an ongoing work about very simplistic model of active fluid, but still what we found seems so interesting and of general importance that I thought this would warrant your attention. So here below you can catch a glimpse of what I'm going to talk about. I hope I'll be able to convey its meaning through this talk. So let's begin. Okay. Yeah. So as many speakers in this mini-colloquia already mentioned, active matter can lead to motility when it is combined with some asymmetric geometry. So each active particle breaks the time symmetry, and if there's some asymmetric object in space, then that leads to current rectification by that object, which leads to force on, which leads to like non-term current and force on body that leads to directed motion and useful work, as nicely illustrated by these wonderful experiments by Kaiser et al. Or Gila and other group. But the question that we want to pose is whether whether we really need such asymmetric body shapes to extract motility. But actually, there are other studies which found that the answer is no. So so let me introduce some previous works first. So here is this work by Mike Katz group, which is about a droplet of active polar fluid, which is in two-dimensional system. And so this droplet is surrounded by a Newtonian fluid. And since this droplet is polar, has a polar order, you can see this active gel aligned in some direction. And then this gel actually turns out to have a splay instability. So as shown by this arrow, it eventually develops some deformation, which leads to asymmetric shape of the droplet. And then this droplet is actually propelled in one direction due to this instability. Now, the deformation can occur in the other way around, and then in that case, the droplet will propel in the other direction. Now, this is about an active droplet, but also you can think about a passive particle immersed in an active fluid, active gel. And in this case, this active fluid has a nematic ordering. And if this passive particle starts to move, then the nematically ordered active gel around it is deformed in such a way that actually this passive particle gets a drag force in the direction of its motion. So this eventually can lead to symmetry breaking motility. Now, then we also have a passive disk immersed in active polar fluid as well, which also leads to symmetry breaking transition. But in this case, what is particularly interesting here is that this system can have can exhibit discontinuous transition even. So in this case, the condition is that you have this active polar fluid surrounding this passive disk. So that you need to put this polar ordering, and then this gel should be tangentially anchored to this disk boundary, so that when you get sufficiently strong extensor active stress, then you can see a discontinuous transition as experimentally verified by this high hysteresis loop that is shown by their data. So I'm not really going to the details about these studies, but what is important here is that these previous known examples of symmetry breaking motility induced by activity require the existence of an active gel which is already ordered. The gel should have some polar order or nematic order to induce activity to induce motility. And what we are going to show here is that actually motility can be induced by symmetry breaking via in an ideal active fluid, which is actually not an active gel, and it doesn't have any polar or nematic order. So even a poorly disordered active fluid can lead to symmetry breaking motility. And we'll show some of our numerical findings, which are corroborated by our derivative approximation theory. So here is our model, which is very simple. So on the left, you can see this diagram showing the run and tumble particle dynamics, which is a very simplistic model of bacterial E. coli dynamics. So the particle moves in some direction at uniform speed u, and then it switches direction at a tumbling rate alpha over 2. Now, and then we put those run and tumble particles on a 1D ring, and then this 1D ring is a very large ring, so length L goes to infinity, but there is a symmetric object immersed here, which is modeled by this triangular potential. So you can see the shape of the potential roughly is illustrated here, and there are active particles, random particles scattered around. And then, so this is a triangular potential. So basically, if you're on the left side of this object, then you get a repulsion of uniform strength minus F to the left. And then if you're on the right, then you get the same repulsion in the other direction. Okay, so this model is very simple. Now, given this system, then you can of course model the dynamics of each active particle, which is just given by this standard run and tumble equation motion. So and then we also give dynamics for this object. So basically, this object is overdamped, and then it is pushed around by run and tumble particles according to this equation. So by the way, this object motility so it needs to be some smaller than the particle mobility. That's what you assume in the end. And then the question is how this object moves in a non-equilibrium steady state. So let me first show you some numerical results from this model. So what you can see from our simulation is that so here the run length of this run and tumble particle is tuned in such a way that it is comparable to the size of the width of this object immersed in this run and tumble particle fluid. Now, what I'm showing you here is like on the right, is a scatter plot of the rescaled object velocity. So here you can see that this object velocity x dot is divided by the active particle run and tumble particle velocity. So like if it is 1, then the object velocity and the run and tumble particle velocity is the same. And on the horizontal axis, you can see this rescaled Wall repulsion. So basically F was the repulsion between active particle and the object, but then I divided by the proportion force, self-proportion force of each running-toward particle. So well, if it goes to 1, then these active particles cannot go over the wall, over the object. So to get any meaningful result here, actually you need to confine, you need to focus on the range where this F is smaller, F is less than this U over mu, so that this value is between 0 and 1, so that these particles can go over the wall, in fact. Now, what you see here is that the scatter plot of this object velocity shows this bifurcation pattern. So when the wall repulsion is too high or too low, you see this object not really moving at all, just moving around this because of some noise. But then in the intermediate range of this wall repulsion, you can see that suddenly the object shows some bifurcation, some kind of symmetric breaking transition, and then it can show a finite value, or rather large value of steady state velocity. So yeah, we are clearly seeing some signs of symmetric breaking transitions as the particle object repulsion is varied. Now let's take an even closer look to this range of data. So here I'm showing you some results we obtained after averaging over many samples. So the color here is representing the number of particles with the stimulation. And actually, I didn't write it here, but this is the rescaled force that was also the horizontal axis variable in the previous slide. Now here, this is the absolute value of the object velocity. And you can see that around like 0.23 of rescaled wall repulsion, as the particle size becomes larger and larger, this object velocity is starting to develop some kind of step-like shape. Okay, so this seems a bit like indicating a discontinuous transition. So we actually decided to measure a quantity called Binder cumulant, which is often used in the analysis of phase transitions and critical phenomena to see what kind of transition is occurring. So basically, what you see here is that this quantity called Binder cumulant is showing a sort of dip here as the particle number is increased. So higher and higher particle number begins to make this curve show a dip to the negative side. So what we expect is that maybe if we continue this trend, then maybe if you use a larger number of particles, then eventually you will see this kind of dip, which is way below the negative side. And this kind of signal is can be interpreted as a hallmark of discontinuous symmetric breaking transition, because this is the property of Binder cumulant. So so far I've given you our numerical test of our numerical results. So we can see that there seems to be some kind of symmetric breaking, which is discontinuous. So questions that we can ask here are, what is giving rise to this kind of phenomena? So we decided to theoretically study this system using a rather simple approximation, which called adiabatic approximation. So we assume that now the object isn't moving because it's pushed around by running into more particles, but we assume that this object is moving at a constant velocity. So basically we are pulling it at a constant velocity through this fluid of running into more particles. And then in the object reference frame, we will assume we can see that each running into particles obeys this familiar stochastic equation of motion. But what we assume here, what we introduce here is this velocity of the wall, which is on the right hand side and highlighted here. So basically we are in the reference frame of the object. So basically the velocity of the object effectively contributes to the force felt by the running into particles. So that's the thing. And then we can move on. So we can move on to obtain a master equation, like formula for the mean densities of one atom particles at different locations. And then, so this is written in terms of one atom particles moving to the right and one atom particles moving to the left. But then we can translate it into another version about one particle density and like the polarization indicating the average orientation of one atom particles. So these are all just mathematics. But what is important here is that this model is so simple that actually people have already found out how to solve this kind of system. So for more information, you can take a look at this paper by Ludosa, Majumdar, and Shea, published this year. So using that method, we can calculate the steady state density for any values of our parameters. And then using that, you can calculate the force applied by these one atom particles on this object by evaluating this integral. Now, then finally requiring that the forces on the object be balanced by like the force applied by these one atom particles and the force applied by this drag force should be balanced. This friction between this object and the medium should be balanced with each other. Okay, 5 minutes. Great. So here is what we can show from our theory. So with the particle as pulled at a given velocity, we can basically theoretically calculate the force that is to be experienced by the one atom particle, the force that is to be experienced as object due to the one atom particles. And then the theory and simulation shows a nice agreement here. And then, as you can see here, the force is positive, which means that it's in the direction of motion. So there's some negative drag force showing here. So basically, if you look into the density of one atom particles around this object, then what you can see here is that while this object is moving to the right, you can see a huge number of one atom particles accumulating in the rear of this object. So basically, these guys are pushing this object forward. So now you can see the what these figures were depicting in the beginning of this presentation. So while in the passive fluid, the particles tend to accumulate before this pushed object. In the active fluid, the particles tend to accumulate behind the object. So that's the difference here. Now, since I'm running out of time, I just give you a very hand-waving argument with the negative drag force here, why it appears. And then what we know about active fluid is that whenever there's a wall, then the active particles tend to push against the wall. So there is some kind of effective attraction to the wall created by the active particles. But then when the body starts to move, then because of the effective force, fictitious force introduced by this body motion, then in the body frame of reference, then there's this force in the direction opposite to the direction of motion. Then if you think about what kind of forces these active particles are feeling, then you can see that on the active particles may tend to accumulate on the left-hand side than on the right-hand side. And also the shape of this wall, which looks like some asymmetric like this, also gives you some idea about what kind of rectification this object will really have on one atom particles, and that also means there will be more particles here than here. So that's the intuitive reason behind why this kind of negative drag force is arising. Now, based on these theoretical predictions, we made predictions about what will be the terminal velocity of this object if we let it move around. Then here's our diagram. So basically, what we can read from this diagram is that as we change the world repulsion on the horizontal axis, then the stable velocity change from 0 to some non-zero like this. So if the run length of the active particle is relatively comparable to the world width, the object width, then this kind of continuous transitions we will get. But if the run length becomes smaller, then what you get is these discontinuous transitions. that you can read from the diagram. So to summarize our predictions by the theory, random particles in the symmetry breaking modality when their run length is sufficiently large enough. That might be because random particles need to sense the effective asymmetry of the object before losing memory by tumbling. And then there's an intermediate range of War repression required for symmetry breaking modality. So that you can see by the transitions occurring here and here in this intermediate regime. Now that might be because high recursion makes it difficult for the random particles to cross the object and experience both halves of the object. And small recursion hardly affects the random particles at all. So that's why you need intermediate values. And both continuous and discontinuous changes is impossible, but we are not as sure about the precise criterion for both types of transitions. So here are some of our future directions that we are trying to pursue. We want to derive a lambda theory, which will give us more precise conditions for both types of transitions. And then we also want to study the fluctuation effects at finite size. And maybe this model is so simple that maybe we'll be able to get some thermodynamic features which were studied by, which were also explained by Patrick Pezonka yesterday and other people. And also it will be interesting to check how these results generalize to higher dimensions and impenetrable objects or systems with hydrodynamic interactions. So thank you for your attention. And I'd be happy to get questions.
Thank you, Janju. Has been really interesting talk, and we have time for just one quick question because it's time of the next speaker. So, any question? I don't see anyone, so I will ask you a question. Have you tried different shapes for this object? So. This triangular shape. So instead of triangular, make it half moon or something like that.
So we tried harmonic one as well, which seems to show similar behavior, but we didn't analyze it theoretically yet. So yeah, that we did not show. But this mechanism is so simple that I believe the word shape will not be that important, not be an important factor here.
And the size of the
size
of this triangular. I mean, not so for another different shape. How relevant will be the size? Same as here.
Size. Yeah, I think the general trend will be the same.
Okay.
So it needs to be small enough compared to the run length.
Okay.
Of the active particle. Yeah. Thank you.
So thank you very much. And we jump to our next speaker, which is Vasco Brad from Lisbon University, and he will present us today his work about enhanced propagation of motile bacteria on surfaces due to forward scattering. So the floor is yours, Vasco. Are you here?
Yes, sorry.
Okay.
Sorry. Let me just share the screen. Okay, can you see my screen?
Yes, perfect. And your pointer too. So go
ahead.
Thank you.
So I would like to thank the organization for organizing this event and also for the opportunity to speak here about this work, which is about the mobility of bacteria in complex environments. I would like first to start to motivate this kind of studies just by saying that this is a way to gain a better understanding of how biofilms are formed, which in itself might be a way to design techniques to prevent bacterial infections, but also to try to design new ways to sterilize medical material, for example. Another application that has been gained some interest from some time now is to try to design our own artificial microswimmers inspired by the ones that we see in nature. And these microswimmers might help humanity performing several tasks, for example, in the cleaning of oceans, but also in non-invasive medical treatment. Here we have many applications. For example, targeted drug delivery, where it might be possible to deliver a drug to a specific spot of our body without harming other parts of our organism. And so I'm going to talk about an experiment that was performed in the University College London by the group of Giorgio Volpe. And they used the E. coli, which is a very famous model bacteria that has already been talked about today. And the E. coli has this funny characteristic: when it's made to move between a surface, such like a glass, performs this kind of circular-like motion that we call chiral motion. And so what they did was to enclose the bacteria between two glass surfaces, and then to test what is the effect that obstacles might have in the environment, and to try to simulate a more realistic environment, they put some obstacles in the way of bacteria. The separation between the two glasses is about 3 micrometers. So basically the bacteria is made to move in a quasi two-dimensional environment. These are some snapshots of the experiments. We can see right away that putting some obstacles, here we have 3 different configurations. Rho here is the density of obstacles, and we can see right away that adding some obstacles to the environment in fact alters the kind of motion as it is to be expected. Here in the absence of obstacles, we see the kind of chiral trajectories that I was mentioning before. So to try to quantify these differences in the motility of the bacteria, the experimentalists defined this circular region of analysis. And then to try to quantify these differences, they defined 2 parameters. The first one they call the effective distance. So the bacteria enters the circle, performs some kind of path, and we actually don't care for the specific path that the bacteria takes, and we only take into account the distance between the entering and exiting point in the circle. And so this was the first parameter that was measured. Then, since this first parameter doesn't take into account the time that the bacteria spends inside the circle, a second parameter was measured, which we call the effective speed, which is just this effective distance divided by the duration of the trajectory inside the circle. So something very interesting happens when we look at the results. So in the left, what we see is this effective distance, the average effective distance against the density of obstacles. And what we see is this quite surprising non-monotonic behavior, where there is an optimal amount of obstacles that in fact enhance the propagation of bacteria through space. This is even verified for higher densities of obstacles. And then of course, if we keep adding obstacles to the environment, then we see a decrease of the proliferation of the bacteria. More interestingly, when we look at this effective distance, what we see is the exact same kind of non-monotonic behavior with the peak, once again, around the 2% density of obstacles, meaning that not only the bacteria are able to travel larger distances, but also that they are able to do it effectively faster. So, and to try to understand what is really happening with the bacteria trajectories, what was done was to measure then a third parameter, which we call the effective propagation direction. Here, this just accounts for the difference between the direction of motion when entering and exiting the circle. So the difference in the angle of between the trajectory and the tangent to the circle. So if this parameter is 0, basically the bacteria enters the circle and leaves at the diametrically opposite point. So it's practically a straight line. And if it is around 180, we see this kind of U-turn that we see here. So in the absence of obstacles, due to the chiral nature of the motion, what we see is mostly this kind of U-turns when the bacteria enters the circle. So it gets back on its own trajectory. As we start to add some obstacles to the environment, namely for the 2% case, what we see is that some more linear-like trajectories start to appear. Of course, once again, if we keep increasing the number of obstacles, we increase the probability of these collisions with the obstacles. And so the bacteria, once again, start to get back on its own trajectory. And so if we look closer at the experiments, there seems to be 2 kinds of interactions with the obstacle. The first one that it is to be expected are the collisions. So the bacteria collides head-on with an obstacle, and then it takes some time to change its direction to a direction that points away from the obstacle. But we also have what we call this kind of forward scattering interactions. In here, the bacteria passes tangentially to an object, and there seems to be some rectification of the circular-like trajectories. Okay, so now, by looking at this, we have an hypothesis, which is to try to answer, we want to try to answer why this non-monotonic behavior happens. And our hypothesis is that there exists an interplay between these two mechanisms, the collisions and the forward scatterings. To try to check our hypothesis, what we did was to come up with a very simple numerical model in which we tried to keep the minimum physical ingredients as possible and still have reproduce the non-monotonic behavior. And to do so, we assume that we have two-dimensional chiral active particles. We say that the obstacles have the same size of the bacteria and are distributed at random. We say that the bacteria-obstacle interaction is described as a superposition of three contributions. So to model the collision type of interactions, we use a repulsive interaction where R here is just the distance between the bacteria and the obstacle. R0 is the radius of both the bacteria and the obstacle. And we also include this random reorientation upon collision that we call tumble collisions. Basically, when the bacteria collides with an obstacle, if the velocity drops below a certain threshold, the direction changes instantaneously to a direction that points away from the obstacle. Lastly, the most important part, to try to model this kind of forward scattering interaction that we were observing, we say that the angular velocity of the bacteria is omega 0, that we take from the experiments, from the average value of the angular velocity of the bacteria in experiments, and we say it's 0 in the absence of obstacles, and it drops exponentially when approaching an obstacle. And so to actually get the trajectories, what we did was to integrate the very famous Legendre equation that says that the forces acting on the particle, the bacteria, consist on a direct term due to the fact that the bacteria is moving against the fluid. We also have the force acting on the bacteria due to its closest obstacle, and we neglect all the other obstacles. We only consider the closest one. And then, of course, we have an activity term where U here is the unitary vector that defines the preferential direction of motion. Since we are in a low Reynolds number regime, we can assume the overdamp regime, and then we get the equation for the velocities and also for the preferred direction of motion, here highlighted, where we have two terms. The first one tries to mimic the collisions between the random collisions between the fluid molecules and the bacteria, so it's just a stochastic term. And then we have this ingredient, which is, as I mentioned, the equation for the angular velocity that depends to the distance of the obstacle. Okay, so when we make our computers to work, this is what we get. Once again, we can see that we have this kind of chiral trajectories in the absence of obstacles. We can see that as soon as we put some obstacles in the way, we start to see this kind of rectification of the bacteria trajectories. And of course, once again, if we have too many obstacles, the bacteria starts to be localized. And so the next step, obviously, is to try to compare the results that we have in the simulations with the ones obtained in the experiments. And here we did three things. First, we considered all the three contributions of the interactions between the bacteria and the obstacle that I mentioned above. So we have the repulsions, the forward scatterings, and the tumble collisions. Just to recap, the tumble collisions is when the bacteria collides with an obstacle and immediately changes its direction. And what we see is that we have a pretty close agreement between quantitatively and qualitatively between the experiments and the simulations. But more interestingly, if we then turn off these tumble collisions, we see that although the quantitative agreement is not as good as before, we can still get this non-monotonic behavior, meaning that these arise from the competition between the repulsion interactions and the forward scattering kind of interactions. Then if we turn off these forward scatterings and we only keep the repulsive part of the interactions, what we see is the monotonic behavior, where the effective velocity and the effective distance decrease as we increase the density of obstacles. Here is just to show that we also have a good agreement with experiments when we consider the third parameter. Then what we did was to use the possibility and the advantage of the simulations and to check if this is a robust result. And so to do so, what we did was to change the distribution of obstacles. So what we were doing before was to place the obstacles at random. Now what we are doing is testing for a periodic lattice and the random trimers. Here in the periodic lattice, what we did was to choose a lattice spacing that makes the lattice complete at the 12% density of obstacles. And then when we decrease the density of obstacles, we take obstacles at random. Okay, since I don't have many time left, just here just to show that these results are robust qualitatively. This meaning that the non-monotonic behavior is not specific to the kind of distribution, obstacle distribution that we observed earlier. Well, and then to conclude, we saw that the experiments show a non-monotonic dependence of the motility of the bacteria on the density of obstacles. We developed a numerical model, and we could show that the non-monotonic behavior arises from the decrease of chirality in the vicinity of an obstacle. This model is also able to show that the behavior is universal in the sense that does not depend on the specific details of the experiment, such as the mechanism of self-propulsion of the bacteria, the specific kind of bacteria, or the distribution of obstacles. And now we have a model that can be used to test the behavior of chiral active particles in a systematic way that might be hard or at least expensive to do experimentally. So to finish, I would like to thank all the people that I've worked with in this project, and to thank you all for listening. If you have any questions.
So thank you very much, Vasco, for keeping on time, and very nice talk. So we have time for questions. If you have any, please write in the chat, and I will indicate when you can ask the question. So I see that we have one question from Raul Marace. So go ahead, Raul.
Yeah, hi, very nice talk. So I'm just wondering, even if you have a random arrangement of the obstacles, how does the motion enhancement or reduction depends on the average number of neighbors a given obstacle may have, even if it is random? So you define a certain length, in that radius, it can have n number of neighbors.
Well, we didn't do a systematic study of that, but we can see, for example, here, if we have the random trimers, what we see is that since the area occupied is not as uniform as in the case of the absolutely uniform distribution, what we see is that there is a larger enhancement of the forward scattering kind of interaction. So the fact that if the obstacles are more spread into space, the probability of collision increases, and so this mechanism takes over the enhancement that the forward scattering has.
Okay, thanks.
So we have another question from Richard Campuzano Cortez. He's asking about what bacteria is recommended to use in the laboratory, and which bacteria have you used to compare your simulation results?
So the question is about what is the bacteria that would show this kind of behavior, right?
Yes.
So the bacteria that was used in the experiments, I did not do the experiments. They were done by Dr. Giorgio Volpe's group. The bacteria used was the E. coli, but this kind of chiral behavior is known in many organisms. To observe this kind of enhancement, the ingredient that we need is that these bacteria need to be flagellated, like the E. coli. The E. coli has this kind of flagella that arrange in a screw-like manner, and so when they rotate near a glass, they do this kind of circular motion. And the same thing is what happens with the obstacles. When they pass near an obstacle, what happens is that they feel the same kind of force that produces this circular trajectory, and they get away from the surface. So what really happens with the obstacles is that the same kind of hydrodynamic interaction makes them get away from the surface, and so the trajectory starts to be more linear-like.
Yeah,
so,
that's
so we need this kind of flagellated bacteria.
Yeah, that's my other question. You don't expect this behavior for other type of swimmers, like you need to this alignment with the obstacle, right? Because
yes.
I've seen that other bacteria tend to get swimming around obstacles and.
Yeah, this also happens with the E. coli. Here, what happens is that if you increase the number of obstacles, you would get this effect that you are talking.
Oh, I see.
And if you decrease too much, since this is also another dynamic effect, if you decrease too much, the strength of this forward scattering will also decrease, so you will not see this effect. But as the simulations show, what you need is some mechanism. It might be the same or for the E. coli. In the E. coli, we know it happens because it is a flagellated bacteria, but in any system, if you have this kind of collisions and some mechanism that rectifies the circular-like trajectories, you in principle would observe the same kind of behavior.
And I like a lot your implicit model, but why don't use an explicit hydrodynamic model to capture how it aligns with the obstacles?
Yes,
because it's all hydrodynamics, right?
Yes, in fact, the details of the interactions are all hydrodynamics, but what we were trying to answer was why this non-monotonic behavior happens, and what do we need? What kind of coarse-grained model can we use to explain?
I understand.
Of course, if we want to capture all the detail of the experiments, we will need to go into much more complex simulations. And
and this answers the question. No, I like coarse-grained models better than explicit everything. Okay. So we have one more question from Oscar. Can you ask the question yourself, or do you want me to read? I can read it. Oh,
okay. You make mentions of putting obstacles on the way of bacteria, but what about soil or nutrients? Did you make any simulations or experiments for the movement in that kind of scenario?
What? Sorry, I couldn't understand because the connection started to break a little bit. Can you repeat the question?
You, besides obstacles in the way of the bacteria, did you make any simulations or experiments for if take sources of food, how the bacteria when there is of food?
No. I know that in the experiments there is some kind of nutrients in the fluid that make the bacteria actually be active, so that they can draw energy from the environment and convert it in self-propelled motion. In the simulations, we assume that this is homogeneous, so we don't consider specific spots of nutrients. No.
Okay, thank you.
So thank you very much, Vasco. And
thank you.
We move to our next speaker, that is an invited speaker, and it's Alfredo Alexander Katz. We thank him a lot for being today here because he's getting up really, really early. He is in the US, so it's like 5 a.m. there. So Alfredo, thank you very much for being here today, if you are here. Which maybe...
I am here.
You are here. So thank you very much, Alfredo. Alfredo is going to talk about self-directed self-assembly today. But I think that it's going to be theoretical, but he do theory, simulations, and experiments, which is not very common, but really appreciated. So the floor is yours, Alfredo. Share your screen and enlighten us. Thank you very much.
Juan, muchísimas gracias. Thank you very much. It's a little bit early, as you can see. The light is barely starting to come out over here.
Very convenient position.
But yeah, it's a pleasure. I wish I would be in Spain eating some jamones with you over there, but anyways, that's life. Okay, so I'm gonna start sharing the screen, and I'll set off from there. Okay. Okay, so yeah, so I also would like to thank Laura and Juan and all the other organizers for the invitation. It's really my pleasure to give a talk here, and we're getting used to this Zoom talk, so it's probably not the worst. We could have been more difficult situations, but okay. So I'm going to be talking about something that we call self-directed self-assembly, and in a second, I think I'll clarify a little bit what we mean by this. So the main inspiration for our work, it's coming from the natural. It just need to move things around. Yeah, the main inspiration for our work is coming from nature, and and there's some really incredible examples and at multiple scales of where self-assembly in one way or another are being used. And obviously the rules are quite different because the interactions, you know, the origin of the interactions come from very different places. But you see things that can happen all the way at very small length scales. And I don't know if you get to see this, but you know, this is 5 microns. And in these little trenches here, you see, you know, you essentially are seeing, this is a diatom. And you're essentially seeing, you know, the lithography of the 90s, but done like a billion years ago. On the other hand, if you can go to very large length scales, and you know, clearly the interactions are not the same, and the rules are not the same, but still there are rules, and that leads to emergent behavior where things, you know, like these vortices start assemble, and that's something that's been studied recently quite a bit about how this active materials, and I think in this symposium that's also been a highlight of an area of highlight. So today I'm not going to talk about active stuff. What I'm going to talk is more about you know, the assembly as is done in this diatom, because it's really interesting. It's formed over time, but it's what I guess biologists would call something like development. But in our case, we would like to be able to see we can achieve something like that with some much more simple rules than you know, than all the complex machinery that biology has. So what I mean by that is, so these, for example, these diatoms are formed over time, and this, what we're seeing here is actually the biomineralized part of it, but that was created by the essentially the deposition of proteins and ions, and these proteins start forming structures that were directing the arrangement of these ions, and little by little, as time goes on, you start forming this kind of structures that, at least for me, they're really beautiful, and it's impressive how such a simple system can actually come up with this. And then there's like a myriad of different diatoms or different biomineralized systems, the corals and so forth, and it's really intriguing if we could actually do this. One of the reasons why we're quite interested is because This also takes the problem from a 2D system to a 3D system, or from, in this case, it takes it from a 0-dimensional system to a 3D system. And that's something that we would be looking for in the future. It's something that has quite a bit of importance, actually, technological. So, so the system that we would like to use to do something like that and eventually obtain 3-dimensional controlled assemblies, it's the case of block polymers. So this is an amphiphilic molecule. This is probably the simplest type of molecule that you can think of that actually starts assembling in different phases without, you know, much complication. Well, I guess without much complication should be taken in quotation marks, but yeah, it goes beyond just hard spheres, and I guess atoms do form all kinds of different structures, but you know, then you'll have to get into electrons and things like that. So this one is just you see it here. It's just I don't know if you can see my point. I'm going to put a no, that's not. I'm going to use a laser pointer. Yeah, that's good. Oops. What's going on? Okay. So I don't know if you see it here, but the so it's basically, you can think about it as a spaghetti, and the spaghetti has two different chemistries. One chemistry here is depicted by red. The other chemistry is depicted by blue. In equilibrium, or at least if you let it fluctuate, you let it loose, it will acquire a conformation that's somewhat like a random walk. But you can stretch this thing out and it will just look like a line. And so some of the things that are going to be important is how long this line is, so how long the polymer is. That's what we call N total. That's the total degree of polymerization. And the other part that's important is this thing we call here the chemical incompatibility. So there's really 3 parameters that will control everything. And in fact, the total one might not be so important at the end of the day, but the relative one of saying how long is the let's say the A block with respect to the total, that is actually important. And this chemical incompatibility is also important. And by controlling those parameters, you can have a very interesting phase diagram where you can go from, let's say if you had very short red, a very short red part, you'll form a BCC of spheres in a sea of the blue, and then you'll move into a hexagonally packed cylinders, some bicontinuous phases called gyroid in this case, and others like perforate lamella or the diamond phase. And then once you're symmetric, you have this thing that basically forms a lamellar system. And of course, there's symmetry across this plane, at least from the theoretical point of view. From the chemical point of view, perhaps it's not as symmetric as this, but you can get pretty symmetric systems, even experimentally. So what we will be trying to do is that we'll be trying to exploit the system that already gives us this very nice range of structures, and see if we can build a little bit like if you think about Legos as if you're building up, we want to see if we can actually build something in 3D where not everything is a cylinder or not everything is a sphere, but where we can actually control what goes in what part and what connects to what. So that's basically what we call self-directed self-assembly. So the basic idea is, and I'll show another slide about this, but the key idea is, first of all, you need to have something that is able to control the structure, at least at the very beginning. That will be what we will call like the seed structure. And then from that seed structure, if you start stacking stuff on top of it, just as we think in the diatom, you know, something started and things are adsorbing at that surface, and somehow that surface is already directing the assembly of the next layer, and then that layer is going to direct the assembly of the next layer, and so forth. So that's what we call here multi-layer stacking. We're obviously working in a simplified system. It's starting 2D. We're going to propagate into 3D, and hopefully one day we will be able to create very well-defined structures in 3 dimensions that do not that are not continuous, in the sense that, you know, this is what we call tailor-made. So they actually follow a precise rule of assembly. So in the old days, and I took this out. I don't know if most of you are physicists in the audience, but in the old days, I used to call this holographic self-assembly. And everybody was asking me, where is a hologram? So I said, well, probably we should take away this holographic word. But the analogy is the following. So in a hologram, you put all the information in 2 dimensions, and then you propagate into 3D, basically through the rules of interference, right? So and diffraction. So in this case, obviously those rules are not the ones that are going to apply, but we're in some sense, we're trying to make the rules such that if I encode something in 2 dimensions, like something like this, you know, how could I propagate that information into a very well-defined object in 3 dimensions as I grow this film? So there are some analogies with holograms in terms of, you know, what is constructive and what is destructive. Here we don't have that kind of interference, but I'll show you later that there are some interesting things that happen, and I think this area begs for more I guess more physics-based theory on how information gets propagated across. And I'll talk a little bit more about this, not a little, actually quite a bit more towards the end. So our vision to do this is very simple. So what we will take is we will take a substrate. We will deposit a polymer film. You know, you anneal it, and you can probably see here that there are some trenches. So the idea of putting these trenches is to direct the assembly at the bottom, and I'll talk a little bit more about that in just a second. But so when you put those trenches, you'll form this, let's say you're forming cylinders, so you'll form the cylinders on the bottom. And then the question is, so now I have this nice cylinders down there, and I'm not gonna touch the film, okay? So I'm just gonna, I know that there's cylinders underneath, but I don't want to develop the film. I'm just gonna leave it as is. And what I wanna do then is I want to deposit another film on top of that. And what I want to use the lower film is, I want to use it as a film that will allow me to direct the assembly of this upper upper film. And as you can see here, there's also, in this case, is also cylinders. And let's say you want them aligned with the cylinders on the bottom. So while you might be thinking this looks completely trivial and this should be like, you know, why are you thinking about that? This process, even as simple as it looks, is not so easy. There's multiple complications, and one of the things that we would like is that the chemistry of the second layer will not be the same chemistry of the first layer. We would like that for a couple of reasons. One is because we can use different solvents so that we don't destroy the first layer as we're depositing. And the second reason is because we might not just want cylinders. We might want something else. We might want lamellae or you know, gyroids, or we might want a mixture of other stuff. And finding the conditions under which this process occurs is not so straightforward. So as you will see in just a second. But this is the vision. It looks simple. It's pretty clear. And then if you can do that, then we think that you could do the next step and the next step and the next step and so forth. Yeah, so there's a few key things that I've been mentioning here. So one of them is directing the order at at the bottom, and that's what we call directed self-assembly, right? So that's where DSA stands for, directed self-assembly. So how do you control the precise order in each of the layers? One of the things I will touch upon, and I don't know how many of you are simulation people, especially in the soft matter area, but I'll probably quite a bit, and I'll tell you a little bit about what our modeling efforts have been in this area. We've encountered some really interesting stuff that for some reason had not been touched before, and we don't know why, but I'll tell you the logic behind how we changed some of the modeling. And at the very end, I'll really go into this information propagation, which is directly related to this self-directed self-assembly. So yeah, so as a starting point, I'm essentially the first thing is that how do you control the order? So if you just dump a bunch of copolymers in a substrate, what happens, let's say this one is forming spheres. What happens is that you form a crystal, but it's a polycrystal. It's all all kinds of different directions. And if you at least I'm trained as a theorist, so if you want to do progress, you at least need to start somewhere where it's a little bit simpler. It has to be well defined what you're trying to look at, and so we need to be able to control what's on that substrate. And in order to do that, what we do is we have this techniques that are called that are all called directed self-assembly, but that depend on a few. So there's a few different flavors of that. So one of them is called chemoepitaxy. So in that case, you sort of you take your chemical substrate, you pattern it, and then you deposit that film on the top. And by correctly choosing which chemical pattern you're going to use, you can get very, very nice order. And these techniques have been really used strongly by the group of Juan de Pablo and Neely in the University of Chicago. And I think this is probably the route that industry has followed nowadays. So for example, well, I'll hold just one second. The other route is to actually put some topography, right? And that's called graphoepitaxis. So it can either be done with posts, as I will show, it can be done with trenches, but that sort of gives you some alignment of where your motifs on your film are going to be, and that will give you some nice order as well. So both techniques are actually currently used. So for sure, I know that if you have an Intel chip in your computer, part of it was made using this particular route, this self-assembly route of graphoepitaxy on something that's called the peanut problem. How do you, which interesting, it's like an optics problem, right? You reach the limit of diffraction of your system. You cannot really do by lithography something, and then you use these techniques to resolve it, because these are chemical techniques to some extent. They're not optical techniques. But I don't know. These are secrets, but I wouldn't be surprised that also some chips are now using these other patterning techniques. So yeah, directed self-assembly is very interesting and can give you very beautiful results. So in this case, we're using graphoepitaxy. These little white dots that you see here are posts that are coming out of the surface. And what you see is that there's a structure being formed inside, and there's a different structure from on the outside. So on the outside, you have what we call the fingerprint face. It's a cylinder face, so imagine like spaghetti, but like you just put it on the surface, it's cooked, and you just press it down, and it will just form some sort of spaghetti face that looks like a fingerprint. But in this area, it's actually very well ordered, and we can actually control what it does in here. So this is a double layer system. There's 2 cylinders on the bottom. There's 1 cylinder coming across. If you start playing with those posts, you can actually change the directions of the cylinders on the top. And so here it goes this direction, here it goes this direction, and here it's going at a tilted direction. Furthermore, you can even bring them, you know, so this was the equilibrium phase. This is clearly not, at least not the bulk phase. It's the local equilibrium phase here. And if you stretch the system a little bit and play with the thickness, you can even make something called perforate lamellar phase, which you know, has these holes. The cylinders here, the posts here also serve as a hole, and it's sort of making a new phase that is not the phase that you had on the outside. So the keys for this are essentially two. One of them is chemical compatibility. You have these posts that are going around, and you need to cover them coat them with some chemistry such that they attract one of the faces of your polymer. Sometimes you don't, you might want to make them neutral. It depends for what you're looking, but that chemical compatibility will have very strong influence on what you get. And the second part is something about what we call commensurability, symmetry, and confinement. So your system is going to try to self-assemble at a particular length scale, and it will find the way to keep that length scale intact and still be compatible with the I guess obstacles, or with the I guess it's not obstacles like in the previous talk, but with these are obstacles, or these are posts, like it will try to be compatible with those posts in terms of the chemistry. So for example, if it wants to wet post and you change the distance between, let's say, row of posts, it will try to keep its inter-domain spacing, that means the cylinder to cylinder distance, constant, but go through all the posts like this. And with that, by just changing the different distance, you can regulate, for example, you know, what is the direction in which the cylinders will assemble. So with these two things, you know, we've achieved really lots of progress, and I can, you know, in 2 dimensions, I can say that, you know, that's a solved problem. We can, you know, it's not completely solved, but it's pretty much solved. We can get all kinds of structures. We're pretty advanced in that area. But going into 3rd dimension really becomes a challenge. So so in that respect, I'm going to talk a little bit about how we are modeling these systems, and you know, some of the, you know, very simple changes that we've done, but that have had a really important impact on. I don't know if it's really had an important impact at the level of as a publication, but for us have had a really big impact on really being able to now simulate complex phases of copolymers. So in the past, and I still work on that. I used to work a lot on something called field theory and self-consistent field theory of copolymers. And but recently we moved to you know, a classical coarse grain approach, which is this dissipative particle dynamics approach. The reason why we did this is because you know, we start realizing that some complex architectures are, you know, take a lot of time in self-consistent field theory. They're a pain to program. And that is just so simple in all these particle-based systems. However, you know, you need a system. If you want to do particle-based systems, you need something that can sample rapidly. You know, you need to sample the conformations rapidly, and in that sense, dissipative particle dynamics seem like an ideal choice. So I'll tell just a very quick story about how this went about. So I had this student, and he started studying this, and you know, when you talk about the bulk, actually, you know, like from the original work, it came in the late 90s. You can really reproduce the bulk pretty well, I would say. But But then problems start emerging when you go to thin films. And the reason why problems start to emerge when you go to thin films, that also gave us quite a bit of intuition about what's going on, is that I call it the entropic contribution of the interface. So when you're when you're in contact with an interface, if your polymer model is not reproducing what you expect it to reproduce in terms of what are the entropy penalties of being at that interface correctly, you will not get the right results. And the clear result that we were getting from the traditional parametrizations of DPD for copolymers, which I think are the traditional parametrizations in general, is that like if you just take a lamellar system, you put a neutral boundary, we know it has to stand up so that this is experimentally validated. And if you use SCFT, you also get that. You barely get it, but you get it. And you get it because in SCFT, to some extent, the chains are infinite. They're continuous, so that the number of monomers, to some extent, it's infinite. But when you're going to a DPD model, you don't have infinite number of beats per chain, and that can become a problem. That actually was a serious problem in this the so-called theoretically informed, self-consistent methods or field methods developed by the Pablo, and they came with a solution that they sort of started changing the springs between the spring laws between the beads, and they would start putting effective spring laws between the beads if they were near a surface, right? So they analytically calculated that, and then they started that. But that sort of becomes a hassle because you have to know what structure is going to be, and you sort of need to analytically solve for that. And so we didn't want to do that. So what we realized that, and I know that this is going to sound really stupid, but what we realize is that if you're doing a simulation of polymers, if you follow the canonical, what's called the Gaussian law for the springs, that means you have a spring between two beads that has zero, that its equilibrium distance is zero. That just doesn't capture the entropy correctly when you're at interfaces. And I'm not going to present that here, but I can already tell you, that law also doesn't capture correctly the phase separation. It gets it qualitatively well and sort of quantitatively well, but it really misses some important points on that area. So so what we decided to do is we decided to put stiff bonds between the beads. They're still harmonic, but stiff, and they have a finite distance. So we moved it from what's called the Gaussian model to what's called the freely jointed chain model. And believe it or not, that tiny change completely changed the way these things behave on surfaces, and now it started to agree with that. I mean, we tried to reproduce a lot of the data that we had from experiments, and it was just impossible with the previous parametrizations. The other things that we've done is we've increased the density. So something's called the density. But that later we realized, it's an important parameter, but it's not the most critical parameter. But we typically work with what in dimensionless units is a density of 5. So that means there's 5 beats per bead space. So these things are actually overlapping with each other all the time. Yeah, so these are essentially the two changes, and we sort of use that and reparameterize what are the interactions that will give you the so-called chi-n's that come through this mapping through this AIJs here. So apart from that, everything just stayed the same. And it's but with this, we were able to, you know, as I was saying, we were able originally also to get this bulk phase diagram, but we were able to start getting things like this, right? So starting, so you're looking from the top, so these are trenches, and what you see here on the bottom are lamellae aligning with the trenches. These are circular trenches, and all kinds of different things that we had experimental information about that we needed to be able to reproduce. And, you know, previous parametrizations were just not able to do that. You know, this is actually a figure from our original paper, and this is done with chemopitaxy. So underneath you have some field that's directing, some substrate that's directing the assembly, and yeah. So we had the structures. We knew that they formed in experiments, and we needed to be able to form them in the simulation. If not, we wouldn't be able to validate it. So we went through extensive validation of this new parametrization. So these things that you see here are exactly those posts that you were looking at in those experiments. Over here, we're looking at some perforated lamellar phases and some new kind of phases that are like pseudo quasi-crystals. So there's a point where you can take these films and stretch them out, and you will actually break the symmetry and up to a length scale. So for example, this, you know, this will be your unit cell typically for one of these systems, let's say this one. But when you start stretching it, and let's say 4 domains come in between 4 posts, there's a symmetry breaking because these systems do not fit. You know, they do not fit, and this is not the symmetry they want to form, and they form this so-called superlattices. So these are rhomboids, and they're rotated 90 degrees with respect to the one on the side. So your real repeating unit cell is this. So they're also called Frank-Casper phases, and that's the one that you saw, this double phase that you saw recently. So that gives a lot of confidence that we can use this to actually study these systems. So what I'm going to now go quickly is into this 3-dimensional self-assembly and show you how we've started accomplishing this, and also some experimental evidence that this is actually possible. So so as I was mentioning, the idea is that you direct the assembly of the first layer, you load a second layer, which now instead of being an AB copolymer is going to be an AC copolymer. We're sharing the same. One of the chemical groups is going to be the same. The other one is going to be different. You let the second one self-assemble on top of this one, and then you ask, you know, under what conditions this is going to be, this is going to allow the information that's contained down here to be propagated to the top. So we've done this in different systems. The one that I'm showing here is a cylinder phase that is covered in a spherical phase, and that then is furthermore covered. So it's a triple layer. So this is a spherical phase, and the last layer is another cylinder layer on top. So we were able to transfer all the information that was coming from the bottom all the way to the top. But that doesn't always happen, okay? So here you have a 3D rendition of how that works. That doesn't always happen. And the reason why that doesn't always happen is because depending on the chemical incompatibility between those 2 blocks, the B and the C, you might get what's called a blocking layer. So the chemical incompatibility of the A with the A is 0, right? So if B and C are very incompatible, like in this case, at the interface between those 2 films, what you're going to form is what we call a blocking layer. So basically your system will want to put A on that surface because A with A will not have any penalty, but once you have a flat surface, right, and that there's no inhomogeneity, basically that's like starting from 0. That's essentially no information. You just have a blocking layer that's frustrating the transfer information from the bottom to the top. as you start lowering that parameter, you find that the best scenario is actually a disordered gas of holes, in this particular case, that transfer the information. And actually, this is related to pinning. So you don't care where those little spots are that are going to pin your system, as long as they, you know, as a set are in the right positions that will actually lead to, you know, to pinning of the upper film structure right above, let's say, this parameter. So this is essentially a gas of holes. If you see this, I don't have a simulation here like a movie, but these things are created and destroyed. So it's really interesting. You're transferring information through a disordered system, but obviously it's disordered. But there are correlations within this disordered system that, and those are the key that need to be maintained. If you make these things too compatible, then what happens is that these films start to mix, and then you again go into a mushed up system, and you lose compatibility. So as you can see, there's some optimal chemistry that has to be achieved in order to do this. So other things that we've studied, and I'll be brief, is this something that was quite interesting, which is this meshes. So how do you make meshes? So over there, before I was just telling you about things that you know, overlay one on top of the other and they follow the same direction. But what can I change those directions? Can I make something that is perpendicular? And we knew from before that if you had some topography, you could do that. But in that case, you needed to develop the first film, and by developing means you need to like basically burn the first film and then, you know, deposit a second one on the first burn one. We didn't want to do that, so we were interested in how we could achieve this, and the way you do it is actually quite interesting, and it's basically by capping the first layer with the copolymer of the second layer. So that's one of the first keys that you have to do. And the second key is that you need to have incommensurability between the layers. So if these are incommensurable, the orthogonal assembly is preferred. And these, so you can see these domains are thicker than these ones, and you start forming these mesh faces. So this here is sort of a phase diagram of that. If they're commensurate, the cylinders that you're seeing from the side, they form along the same trenches, just like this case. But if they're incommensurate, and those are the conditions, you start getting these other more mesh-like assembly. So this is quite interesting, and we wanted to test that experimentally. And very recently, we've been able to do this. So that was the first picture that you saw in the title page. But this is basically the phase that I'm showing you that we predicted before with the simulations, and that was achieved on this is a PS-PVP system, polyvinyl pyridine, and what you're seeing here is now something where we converted things to metal, so you can actually see it with an SEM. But these are layers on the bottom that were deposited, and then another layer was deposited on top, and start forming this perpendicular layers on it. And this is the first time that something like this is achieved, and so we think we're going along the correct routes for this. Now it will be very interesting to see you know, how we can start propagating much more complex information. What we're particularly interested is in connectivity. So we're basically interested in the topology of the network, and that will be something really interesting to see in the future. So I'm just going to summarize about what we've achieved and how we think we can start emulating the natural world, the biological world with these systems, and really it's how do you control the metastable states? Basically here it's all about how do you control those metastable states, keep them stable, and be able to use them and harness them keep on growing something on the third dimension. So we think this self-directed self-assembly is a promising route, and that the propagation of information so far we've achieved typically through disordered interfaces, and what it's clear though is that the correlations at that interface are important. So if we can come up with I think a big question is, are there more robust interfacial designs that will allow propagation in I guess in a very robust way that will absolutely not be broken. So in that sense, we're looking into defects and topological I guess defects such that we can sort of entangle topologically different structures, but that's still it's very difficult when you work with soft matter to do that, and so that's still an area of active investigation. And yeah, the last thing I just want to say is that and I think you'll hear more from us in the future, in the near future, about capturing the correct behavior of the model is critical. Sometimes it sounds stupid, but it's critical, and thin films just reveal that those original parametrizations just were not up to the task for really studying thin films. So with that, I'm going to just thank the people that did the work, most of the work. So in this case, pretty much all the work that I showed you, except the first part, was done by Heijin Huang, who's this guy that's been photoshopped into the picture because he didn't come that day. But that's Heijin, and this is basically his PhD work. Some of the other work that was done is like those weird structures. Yi, Ding, and Karim had been working on that. But I've been blessed. So the experimental part, we worked together with Professor Caroline Ross, and in the past, we've done some work with Professor Carl Bergen. So with this, I'll just thank you and welcome any question that you can have.
So thank you very much, Alfredo. Really nice. I wasn't aware of this work, but it's really, really interesting. So we have some time for quick questions. And I see one from Manuel Martin Bravo. You can ask it yourself, Martin. Manuel.
Hello, thank you for your talk. I'm rather going for a more simulation related question, no? Because I'm more into modeling, and I would like to know more details on the modeling, because before you have mentioned a soft sphere, let's say, model of interaction. But I would like, if it would be needed to add, maybe, if it makes sense, maybe it doesn't, to add anisotropic terms, and also if it would make sense worrying about the role of the tail of the interaction, but it is killed, it is zero. And also, how do you model the interaction with the substrate?
Yes. Okay. So okay. So these are all excellent questions. They're quite a bit. Okay. So let me well now. So what? Let me go back here. I'll use the pencil. Yeah. So let me see if I can. So bear with me. So okay. So so what you're saying, I think you're going for a little bit more of a chemical, for a true chemical sort of description. And let me tell you a little bit what is the logic behind what we're thinking. Okay. So if I have chemical bonds, okay, and I'm just drawing them very badly, but let's imagine these are atoms. So the logic that we have behind our new parametrization is the following, okay? Oops. so the basic logic is this one, is we're thinking that these beads are what are called Kuhn beads or Kuhn segments. So what that means is that we're replacing several atoms with one atom or with one bead. It's a soft bead. Now I'll go in a second why that is. So we're going to replace it by, let's say, what will be one bond now. And the reason why we need to have something more like this is because these are space filling objects. So the density is high. So what you expect is that several of these segments, you know, they don't fill the whole volume of that. They only fill partially one volume. So that's why putting one on top of the other, like these higher densities, make sense. Now, in terms of the tails, what you're saying, like if the tail matters, I wouldn't be surprised that it does matter. And also the anisotropic ones, if these Kuhn segments are actually pretty big, internally, the interaction won't be isotropical. It will be anisotropical. And for the moment, we've avoided that because the kind of polymer that we've been working with is they're pretty flexible, but I bet that if you start working with a little bit more rigid polymers, that would become important. So yeah, so the basic logic is that each of these beads that you see here, it's taking like about 10 atoms of a real chain, or it's taking the typical length, the correlation length between 10 atoms of each chain. And then because there's multiple chains, they're all sort of going to pack in that same space. There might be 5, 10, depends on the, you know, with DPD you can put whatever density you want. It just depends how long you want to wait. And yeah, so they will share this space, and that's where this repulsive, and that's the simplest term that we can think of. It's that repulsive Gaussian force to do this. But where the key issue lies is that when you're at the interface, so then the question was, how do you model the interface? So the interface, we actually model it by beads as well at higher density. So they're very high density. So we put many beads on top of each other, and that just creates a wall, but the repulsion is pretty similar. Now, so what we believe is happening is that when you're at an interface, the entropy of a chain, of a freely jointed chain and of a Gaussian chain are only equivalent in the limit that that freely jointed chain is enormous, super long, okay?
But if the substrate is of the same kind of the coarse grain, wouldn't that favor maybe some patterns in the results? Or because, for example, if it was modeled as a perfectly flat surface instead of being built of the same kind of coarse grain materials, maybe the patterns would be less likely to take place, or I don't know.
What patterns? We don't put a pattern. We put a surface that has. I would say this is a pretty flat surface. So we have 5 times more the density that we use on the bulk. We let it stabilize, and I believe this is going to be really, obviously it's not going to be perfectly flat, but it's going to be pretty flat.
Okay.
I actually think it's the entropy. I think these two things are equivalent, but only in the limit that the chain is extremely long, and that limit is well attained with self-consistent field theory. My opinion is that limit is not well attained when you have 20 bead chains, And because you need 10 to reach, like, it's like if you had 2 Gaussian bonds, right? In this case, 2 real Gaussian bonds. And in experiments, you don't have this is going to sound actually weird, but in experiments, you are barely going into that Gaussian regime. So because you can, like, I can give you numbers. So we know that there are correlations about 10 monomers along, right? In a typical, let's say, polystyrene. So the molecular weight of polystyrene is 100 of a styrene. So if you have 10, you're talking that it's 1,000. So every bead here has, in some sense, a molecular weight of 1,000. I guess it's in grams per mole. And your typical polymers that we use is 20,000, right? So you basically have 20 beads, okay, and 20 real beads, and these are all correlated. So that's not going to be a Gaussian chain. The Gaussian chain is going to start after you have 10 of these effective bonds after. So that's where we think is the key. And I am also guilty of this. As a theorist, I thought, oh, this Gaussian chain is perfect. That'll do it. But I think in order to make real comparisons with experiments, it's not bad to go to coarse grain, but I think we miss some of those physical insights or chemical insights, physical chemical insights in the materials themselves. I don't know if this answered your question.
Yeah, thank you.
So we are running kind of late, and I would like to make use of my position as a chair to ask a question. But before my question and end up the conference, can we take a picture so everybody can switch on their cameras? So we take a picture. And Laura, you will do it?
Yes. Alfredo, if you can unshare your screen just for the...
Oh, yeah, that's true.
Thank you. If everyone can turn on their cameras, thank you. Yeah.
And smile.
Can you fit everybody?
In 2 pages, but it's fine.
Okay.
Thank you. This is done.
So a very quick question, Alfredo. The magic happens with these holes in between the 2 faces, right? For patterning the top layer. So there is a critical concentration of these holes. It's a sharp transition between a homogeneous layer and a kind of Gruyère layer.
Okay, that's yeah. Actually, that's an excellent question. It's not super sharp, so it's not like you get it and you don't get it. No, it's not that sharp, but it is. Let me see if I have it somewhere. I don't think I have it here, but it is somewhat pronounced. That compatibility. Well, the Gruyère you get, but it's not the density of holes in that Gruyère layer. I like that term, the Gruyère layer. We're compatibilizing this through Gruyère cheese. Yeah, the number, it does matter. So because you can make a lot of holes and then it starts to break. Also, those holes, if that becomes a phase, that phase might actually be incompatible with the lower substrate or the upper substrate and might start giving you noise. On the other hand, that might be something to use. But yeah, so I see it more as so far we see it as a pinning. And this is why I say it's not robust yet, because it only happens under certain conditions. If we could have something that, it's forced to happen no matter what, that would be much more ideal.
I like the idea that it's kind of dynamic, right? That you make a hole, it reforms and grow in another place. I think that that's kind of nice. So.
Yes, it's just not very robust, but yeah, it's interesting. It's just it's basically you have a gas of holes and the correlations between those holes, because they're not, you know, they look random, but they're not really random.
Yeah.
That is what's actually transferring that information upwards. So it's transferring that correlation.
So really cool. Thank you, Alfredo. And thank you all for participating in this mini-colloquia. I really enjoy it, and hopefully you too. And thank you very much for sending us this ton of contributions we received. In fact, we had to add an extra day to fill up all the participants. And thank you the rest of the organizers, Laura, Felix, Timon. It has been great working with you, and I would like to give you the word if you are interested in saying something. Felix, Timon, Laura.
Yes, I think it was a very interesting experience. Of course, doing things online is not as doing things in real, and there is a gap in that, that's for sure. But I think the experience was quite interesting. All of us are quite new to this way of interacting and exchanging ideas and discussing. So I am very satisfied about how it went. I could not attend all the talks, and I regret that, because some part today and also some of them on Wednesday. But I think the result was, to me, quite satisfying. I think you did a pretty well job, you, Laura and Juan, in running these sessions, at least in running these sessions with all the software and the Zoom video details and so on. You did pretty well, so I wish to thank you, and also Timon, in all the process that we had in selecting also the speakers and the contributions. I want to join my congratulations and thank you. Thank you, and also the participants and the speakers, for sure, which made it possible. Thank you very much.
So, Timon.
Thank you, Felix. I can only second you in especially thanking Juan and Laura for managing the whole online experience, where Felix and I could just sit back and receive the emails and approve all the work that was done by the two of you. So thank you very much for that. And thanks everybody for working together and making the conference go on despite everything. So I'm quite happy that we could have this mini colloquium even in such weird times.
I just want to...
No, yeah. I was just saying that we cannot do more than just watch ourselves in the screen. But anyway, I see Margarida, who is there. Hello, Margarida. Because long time we don't see each other. But anyway, thank you all. Laura, yeah.
Yeah, thank you all. I just want to say thank you all. My co-organizers that were excellent, all the speakers and participants, and that's it. I'm now closing the session officially, but I will leave it open in case you want to talk to friends without recording and things like that.